about Richard. He received his degree in biology from Brown and went on to get his Ph.D. at Harvard University studying muscle physiology and provided some important insights regarding the regulation of muscle stiffness as it relates to muscle spindle feedback during his dissertation. Some seminal works that have been very highly cited over the years. He went on to do his postdoctoral research training at the University of Alberta in Edmonton. And then after a couple of years in Seattle at University of Washington, made his way to Atlanta, where he had a very productive line of research in the Department of Physiology as a professor at Emory. And then in 2007, some individuals here at Georgia Tech convinced him to come on over to Georgia Tech, and since that time he's been the chair of the Department of Applied Physiology here at Georgia Tech. And so Richard has received numerous honors and awards for his research, and probably what he's most proud of is for his mentorship, which I can tell you is a wonderful mentor across the spectrum of undergraduate, high school, all the way to young faculty, and kind of a testament to his science and the impact of his work. Kind of speaks for itself in the history of his funding, which has been continuously funded by NIH since over the last 30 years. And so it's certainly a testament to his science. The main theme of his research has focused on trying to understand the mechanisms that regulate mammalian coordination as it relates to primarily locomotor tasks as well as postural tasks. And some of his important contributions, of which he'll talk today, speak to the importance of proprioceptive networks, and specifically muscle spindle and Golgi tendon organs, and really how they're organized in a way that lends a neural linkage that is certain, as he's proposed over the years, to play a very important role in regulating whole limb mechanics. And so with that, I think that's some of the details of which he'll share with us today. So we're very excited for a very informative and interesting talk.
Thank you, Mark. Thank you for that kind introduction, and thank you everybody for braving the storms to be here this morning. Boy, that's a bright projector. It's a little intimidating to give a talk to your own colleagues, but see what we can do here. This is actually pretty much a brand new talk, so if anything along the way isn't all that clear, just raise your hand. I'm happy to keep it informal. Anyway, we know that the spinal cord has intrinsic motor capacities. It contains pattern-generating networks, which can regulate stepping as well as other kinds of rhythmic behaviors. It also has circuits, neural circuits that arise from receptors in muscles, skin, and joints and so forth, which are involved in the regulation of posture and balance, both during quiet standing and movement. And all of these circuits are, of course, very closely integrated with activity of the brain stem and the brain, but it's particularly interesting to focus on the intrinsic motor capacities of the spinal cord, because unfortunately there are individuals who have spinal cord injury. And so until we find a way to completely regenerate the spinal cord after spinal cord injury, which is a long, long way off, the approach to rehabilitating patients with spinal cord injury has to depend on a deep understanding of these intrinsic motor capacities. So that's what we're going to talk about today. So we're still kind of in the infancy of understanding the details of these circuits. They've been worked out in great detail in lobsters and crabs and other invertebrate animals. Mammals seem to be particularly difficult to, intractable to get to. So I'm going to sort of report on some progress in this light. I want to frame the question about the involvement of proprioceptive circuits, that is, those circuits that arise from muscle receptors, predominantly muscle spindles and Golgi tendon organs. I want to frame the question in the following way, and that is spinal circuitry modular? And the reason for framing it in this particular way is that most of the classical descriptions of spinal circuitry provides us with a modular model of how it works. And this is, of course, a reductionist approach, because if you can understand how a single module works, then it's just a matter of adding more modules, and then you've got the whole system. Easy. Well, it's not so easy, because it's a modular system. You're having to pass the buck of coordination to some other level in the central nervous system. And what we're trying to argue here is that actually a lot of the coordination takes place right there in the spinal cord. So just to give you the classical view, which will provide a little bit of introduction for those of you who are not familiar with this field, this is a classic textbook picture of spinal circuitry that you find, and this is the latest edition of Kandel, Schwartz, Jessel, and others, probably the most famous neuroscience textbook for medical students in the world. And basically, you have here a muscle with a stretch receptor in it called a muscle spindle, and that feeds back to the spinal cord. And so we have the muscle itself, which we can call the agonist. We have another muscle, which we call a synergist, which does similar things. Maybe not identical things, but similar things, and I'll get to that in a little bit. And then another muscle that has the opposite action called the antagonist. And this little circuit was first proposed by David Lloyd in 1946, and it's called the Myotatic Unit. And it's sort of a unitary idea of how the spinal circuits operate. And basically it's pretty nifty, because if you perturb the elbow joint, all of these pathways, the excitatory pathways from this muscle spindle back to the agonist, its synergist, and the inhibitory pathways to the antagonist, all conspire together to resist that perturbation. It all adds up to resistance to the perturbation. So it's a very nice idea, and it was sort of the cornerstone of our thinking for many, many years. Unfortunately, it's very flawed, but we'll get to that in a little bit. So that's the myotatic unit concept, and it's still pretty much taught to medical students and graduate students today as being one of the building blocks of the spinal cord. Another building block is as follows. There's another receptor in muscles called the Golgi tendon organ, which have in-series connections between the muscle and the tendon. These Golgi tendon organs measure muscular forces, whereas the muscle spindle measures the lengths of our muscles. And tendon organs, their afferents go back to the spinal cord, and they have more complicated connections in the spinal cord. And under many conditions, these connections tend to be inhibitory rather than excitatory. And the explanation for the function of these pathways isn't quite so clear-cut as it is for supposedly for the muscle spindles. But to give you some idea of the complexity, the Golgi tendon organ circuits, which are inhibitory under many conditions, under some conditions, particularly during locomotion, scratching, and other rhythmic behaviors, some of these 1B afferent pathways switch from an inhibitory pathway to an excitatory pathway, and they actually excite the motor neurons, causing a positive force feedback loop. And one of the challenges is to understand how these inhibitory pathways and excitatory pathways are integrated and intermixed during natural movements. A third kind of modular model of the spinal cord is this one, proposed by James Houck in 1972 and explicated more completely in 1979. He happened to be my thesis advisor, so I had to talk about this, of course. What he said was, well, we've got muscle spindles measuring length and tendon organs measuring force, but where's the integration between these two? What do they do as an integrated set? Most neuroscience textbooks tend to talk about these pathways as if they were two separate things. And that continues to the present. Well, he made a very, very astute observation. He said, well, if you combine excitatory length feedback from muscle spindle receptors together with inhibitory feedback from Golgi tendon organs measuring muscular force, then you end up with a negative feedback control system which regulates not length, not force, but the relationship between the two. Change in force over a change in length is stiffness. So what he did is propose a hypothesis called the stiffness regulation hypothesis. in which he said that the function of these pathways is to regulate muscular stiffness by integrating length and force feedback together. Now, this model also turned out to be incorrect, but we were left with this very powerful idea that length and force feedback can be integrated at the level of the spinal cord to regulate stiffness. And what I'm going to try to convince you is that this regulation of stiffness does not take place at the level of a single muscle, as implied by this original version of the hypothesis, but the regulation of stiffness takes place at a more integrative level, the level of the intact limb. Okay, so here's the problem with these simple models. As I said before, the idea of a modular system is that all the integration has to take place somewhere else, so you're kind of passing the buck. But in the first place, the musculoskeletal system is not modular itself, the system which is being regulated by these pathways. And here are some of the reasons why that musculoskeletal system is not modular. It's not the simple sort of synergist antagonist system that I showed you in the beginning with the myotatic unit. Take the biceps femoris muscle in the cat. This muscle crosses the hip, crosses the knee. It has fibers that extend all along here through fascia and tendon, and it provides coupling across the hip joint and the knee joint. It also is not shown here. It has fascia which connects these muscles to the ankle as well, so that muscle is actually working at several different joints all at once. The other thing is that this muscle is, because it's on the lateral side of the leg, tends to rotate the shank outward. I didn't do that very well, did I? It tends to do that. And its counterpart, the semitendinosus muscle on the medial side, tends to do the opposite. So this muscle and the semitendinosus are both agonists and antagonists at the same time. They're synergistic for knee flexion and antagonistic for knee rotation. So what do we do that? How do we squeeze that into the myotatic unit concept? So because muscles are multi-articular, multi-axis, and have fascial connections, which makes them have transmit forces in other places than is accounted for by tendons, joints throughout the limb have viscoelastic coupling because muscles have viscoelastic properties. The other reason why the system is not modular is that, of course, our limb segments have inertia, and therefore the movement of any one limb segment is going to give rise to movements of neighboring limb segments by inertial coupling, like this. I'm not using my wrist muscles to do this, right? So we have this problem of intersegmental dynamics. When we're doing something very ballistic and very rapid, how do we gain control over our multi-segmented limbs so as the intersegmental dynamics do not interfere? Not only is there inertial coupling, but that inertial coupling is non-uniform, because the big massive thigh here next to the smaller shank and this little itty-bitty foot here means that the inertial properties are going to be changing with respect to the position on the limb. And we're going to get to that a little bit later with some of Mark's work that I'm going to briefly mention. Just one thing on fascia. This is Victoria Stahl. She was a Ph.D. student in biomedical engineering that worked with me. Did her thesis now as a postdoctoral fellow in Randy Trumbauer's laboratory at Emory. She actually interfered with the fascia between these muscles and the calcaneus and showed not only that there was a loss of acceleration at the ankle joint, but surprisingly found that the animal, this was a decerebrate cat, this is an animal that's been rendered permanently comatose by removing part of its brain, actually most of the brain which is involved in consciousness. These animals walk very nicely on a treadmill when you start the treadmill, and you can do invasive experiments on them without worrying about the ethical concerns because the animal cannot suffer at all. She found that that this is a tracing of during the step cycle of the foot's going back and forth in sort of a little elliptical shape. When you cut the fascia in different stages, the limb sort of loses control and starts going all over the place. So fascia is really an important thing and adds to our story that the musculoskeletal system is not modular, that is, forces are transmitted widely throughout the limb. Spinal circuits aren't modular either. And in fact, as I said before, if spinal circuits were modular, they would contribute little to coordination. This has actually been known since the 1950s, when Sir John Eccles, Nobel Prize winner, and his daughter and Anders Lundberg wrote a series of classic papers which are widely quoted but very seldom discussed in any detail. They pretty much demolished the myotatic unit concept by saying when you actually look at the way feedback from muscles is distributed within the spinal cord, it does not conform to that simple model of the myotatic unit. And I won't drag you through the details, I promise, because we only have a few more minutes. So that was the first thing. We've known that actually for a long time, but this work is, as I said, widely quoted, but very seldom discussed or brought into our modern conception of what's going on. As a side note here, I wanted to remark that a very talented postdoctoral fellow of mine named Young-Hee Chang, that was at Emory, who's now an associate professor in applied physiology, did some experiments in my laboratory at Emory where we were asking a particular question, and the experiments turned out not to be, well, we couldn't answer the question with the experiments we did, and neither of us won the bet. So he came over to Georgia Tech and sat on the data for a year and then came up with this really fantastic idea that he could use the data for, which is that the control of multi-segmented limbs is hierarchical. Task or limb level variables like total limb length, limb angle, so what the whole limb is doing during movement, are controlled at the expense of local or joint level variables like the movements of individual joints. So when you're walking through, when an animal or human's walking along, the trajectory of the limb as a whole is maintained very, very reliably from step to step, but there's a lot of variation in what individual joints are doing, presumably correcting some sort of internal disturbances in order to keep the task level variables. That implies there's some kind of local mechanisms of coordination, and we think those local mechanisms are within the spinal cord. So for all of these reasons, we thought it would be best to try to reinvestigate the question of spinal circuitry. So moving back in time a little bit, two of my graduate students, Stephen Bonacera, who's, by the way, an electrical engineering graduate of Georgia Tech. He did a master's in electrical engineering here. Did a Ph.D. with me and then went on to medical school and is now associate professor at the University of Nebraska. And Ronnie Wilmink, an engineer from the Netherlands, who did his thesis with me and is now a practicing engineer in Marietta. These two then students did some remarkable studies in which they took this decerebrate cat preparation again. Because we could do invasive procedures on these animals without the risk of ethical considerations, the animal was permanently comatose again. They dissected out the tendons of individual muscles and attached them to stretching devices. And by stretching these muscles in different combinations, they could figure out, by recording the force responses, they could figure out how these muscles talk to each other through proprioceptive pathways. And what they discovered, and this is sort of an anatomical rendition, what they discovered is that in agreement with the work of Eccles et al., that Golgi tendon organs in muscles, and the muscle, let's say, would take this muscle here, one of the quadriceps group. The Golgi tendon organs in this muscle, colored in sort of mustard yellow on this screen, the tendon organs project not only to neighboring muscles, but also muscles lower down in the limb and vice versa. So they found that force feedback from Golgi tendon organs is widely distributed through the limb, is not localized and not modular. This served to extend and confirm the results of Eccles et al., but it did something very important. The work of Eccles, the experiments of Eccles were performed in animals that were deeply anesthetized with sodium pentobarbital. So these animals the spinal cord was essentially asleep in these experiments. And so what Eccles did was essentially kind of electrophysiological anatomy. What these experiments did was to say when there's active force production, the muscles are working and the spinal cord is alive and well, that the same findings from Eccles were still still applicable, and they were able to document how these pathways change their activity with respect to the different forces and lengths in the muscle, and I won't go into that in detail here. So this is it gives you kind of an intuitive idea that these pathways from the Golgi tendon organs are widely distributed, but this isn't particularly, this is a nice sort of anatomical illustration, but what we were really interested in doing was trying to find a more quantitative and more comprehensive way of expressing how these pathways were distributed in relationship to the anatomy of the limb. And so these 4 individuals came into the picture, starting with this guy over here, John Henry Lawrence III, was my first graduate student at Emory, biomedical engineer from Brown University. What we decided to do was to do a quantitative analysis of the anatomy of the limb by trying to understand how muscles produce torques about different joints. And John did a very nice thesis where he documented the torque contributions of different muscles that cross the ankle joint of the cat in a full 3-dimensional model, showing that there were muscles that synergistic and antagonistic relationships are not as simple as was once thought. Then I had another very talented postdoctoral fellow named Tom Burkholder, now associate professor in the School of Applied Physiology, who came along from Rick Lieber's lab. And what Tom and I decided to do was, or what I decided that Tom might be interested in doing, because he's the guy with the skills, was to actually build a computational model of the feline hind limb, one that included all of the anatomy and all of the mechanical properties, the inertial properties, and the whole shebang. Once we had a computational model of the hind limb of the musculoskeletal system, then we could start to test hypotheses about what these different proprioceptive pathways would do to the coordination of the limb. And this is a process which is still ongoing. We're not there yet, but I want to give you sort of an update on where we are. Tom then got a job here at Georgia Tech, and he started working with Dr. Ting, and they expanded the model further and put it onto a more user-friendly platform. And then Tom had a graduate student named Nate Bunderson, who just left my lab as a postdoc and to take a job in industry. And Nate has sort of taken this musculoskeletal modeling to its present level of sophistication, which is considerable. He also had help of a number of other students in the Ting laboratory, Lucas McKay, Jeff Bingham, Hongshul Son, for example. And he produced a platform called Neuromechanic, which is this really complete model of the feline hind limb. And actually, he's gone on to model other beasts as well, including the human subject. So I'm not going to be able to go through a detailed description of this model, Neuromechanic, but one thing we extracted from this model became very useful in trying to understand the relationship between the mechanics of the musculoskeletal system on one hand and the organization of proprioceptive circuits on the other hand, to see how they interact. And so, because this model contains a lot of morphometric information, that is, where the different muscles attach to different points on the skeleton, we were able to come up with a way of quantifying the mechanical relationships between muscles in the limb. And this method is actually the brainchild of Tom Burkholder. These anatomical diagrams are nice, but they don't really get you to what we want to get. So what Tom said was, look, we've got the hind limb of the cat. The model is a 7 degree of freedom model. It has 3 degrees of freedom at the hip, 2 degrees of freedom at the knee, and 2 degrees of freedom at the ankle. So what you do is you construct a 7-dimensional space, and the moment arm of each of the 31 muscles that we used in our model, the moment arms of each one of those muscles could be expressed as a vector in that 7-dimensional space, okay? Then what you do is you take the angle between a given pair of vectors, and that tells you how anatomically similar those muscles are. If they're actually pulling in the same direction, they're called highly synergistic. If they point in opposite directions, they're called, you can call them highly antagonistic. If they're at right angles, they're neither synergists nor antagonists. And when you go through this pairwise computation of these angles, you get the following beautiful matrix, which you can use for tiles on your bathroom floor, whatever you'd like to do. So all of the 31 muscles are here on the 2 axes, and each one of these cells provides the mechanical relationship between those muscles in this term. So we have the red ones are the agonist ones, and the blue ones are antagonists. And so that is our representation of the anatomy of the hind limb, essentially. And you can see, we're not going to go through all this in detail, of course, but what you can see is lined up along the diagonal are the big synergistic groups. Let's say these muscles up here are the triceps suri groups, these muscles here in the shank. Down here we've got the quadriceps groups, these big knee extensors up here, and so on and so forth. So these are all lined up as agonistic muscles, and you might wonder what all this is about. The order in which the muscles were listed here on this matrix were determined by a cluster analysis based on similarity. So which muscles were most similar to each other. And by doing that cluster analysis, it actually automatically determined the order in which these muscles were listed. So everything was nicely lined up. So you expect to see lots of red here and lots of blue out here, which is kind of what you'd expect. Although it's not exactly, if the myotatic unit concept were actually the way things worked, the anatomy would be just all red here and all blue here, in very distinct little sort of little columns like that. But you can see there's some blue here in the middle, and some of these blue areas down here are much wider than you would expect from the myotatic unit model. So just take my word for it. This matrix here sort of shows you that the anatomy that the myotatic unit model is based on is a little deficient. Now, if we take this mechanical matrix and pair it up with a matrix describing the distribution of feedback from muscle spindle pathways, which is shown here, it's exactly the same axes, but now the colors represent the connections due to muscle spindle receptors. Remember, the myotatic unit concept was muscle spindle in one muscle excites itself and synergists and inhibits its antagonists. So if the myotatic unit concept were the way things worked, you'd see all red here and you'd see lots of blue down here. We're not going to talk about this in great detail, just wanted to point out a couple of things. There are some interesting connections here, like this one and these down here, which represent excitatory connections between muscles that either work at different joints than the muscle where the spindles are located. That's not consistent with the myotatic unit model. And in some cases, they work these connections are between muscles that span different joints. So this kind of documents the extent to which the myotatic unit model is applicable. You'll notice also that the distribution of length feedback is a little bit more circumscribed than the distribution of these antagonistic areas here. A lot of these blue areas here represent mechanical relationships of muscles that work at about different joints, whereas the muscle spindle pathways, which actually is a little bit consistent with the myotatic unit, tend to be a little bit more circumscribed, but in general are not consistent. And then finally, this is the matrix identifying the distribution of force-related feedback from Golgi tendon organs, again compared with the mechanical matrix over here. We can see a couple of interesting features. First of all, the finding of inhibitory force feedback seems to correspond with those inhibitory regions linking muscles that work about different joints, which is a more quantitative way of expressing what I showed you in those anatomical diagrams, that this inhibitory force feedback seems to be distributed widely throughout the limb. Also, within muscles that have similar actions, we find inhibitory force feedback among some of those muscles too. So one of the goals here is then to try to understand these pathways, and again, it's a little bit beyond the scope of this lecture to go into this in detail, but this is getting us sort of on the way. So I'm just going to sort of tie up where we are with this part of the talk by just going back over the take-home messages. So the mechanical and neural circuits of the motor system are distributed. They're not modular. That's the first big conclusion. The neural circuits, if they're regulating stiffness, and I'll show you some of the evidence for that in a second, are not regulating stiffness on a muscle by muscle or even a myotatic unit basis. They're regulating stiffness on the basis of the entire limb because of these widely distributed pathways. In support of these ideas, and one of the predictions is that this widely distributed inhibitory force feedback would help us solve the problem of these intersegmental dynamics. And in fact, patients with a condition known as large fiber sensory neuropathy, who actually lose the sensory feedback from these muscle receptors as well as skin and other places, have great difficulty with intersegmental dynamics. They cannot control the coordination of their joints no matter how hard they try or how hard they concentrate. So this is actually very nice evidence in favor of this idea that the distributed properties of the musculoskeletal system are matched and regulated by a distributed set of proprioceptive pathways in the spinal cord. As I said, we are now in the process of incorporating the patterns of force feedback and length feedback that I just showed you on those two matrices, of incorporating those pathways into the neuromechanic model, and then we can do experiments on the model and test different ways that these pathways might be distributed in order to see what the implications they are for whole limb function. Any questions at this point? Because I'm going to jump into a slightly different topic now. Okay. Yeah. Yes.
So the mechanical matrix is symmetrically distributed.
But they are not. The feedback circuits are not symmetrically distributed, right?
So right now we're trying to integrate the length feedback and force feedback into that mechanical feedback.
That's right, exactly. Yeah, actually, Amy brings up an interesting point that that mechanical matrix I showed you is symmetrical. The feedback is distributed non-symmetrically, which actually raises some interesting issues with how a whole multi-segmented limb, how its properties can be regulated. The technical idea I'm getting at is that if you have a multi-segmented limb with a bunch of muscles connected to it every which way, and there's no feedback at all, that system will behave in an inherently spring-like way. If you have asymmetrical neural feedback, there is the possibility that that system is non-spring-like and non-conservative. Its technical term is non-zero curl. And under those conditions, it may have some special properties which are not conducive to movement and posture. It turns out that actually there are some cases where non-zero curl is actually consistent, and actually a paper by Nate Bunderson has actually talked about that in some detail. But anyway, so there's the symmetry business. Anyway, yeah, Bob.
So this is largely based on animal models?
Yes.
And do we know anything about how this compares with the human? I mean, are there differences in the human as opposed
Right.
to these animal models?
Yes. So in the human, you can't do the sort of experiments we do in the animals, right? So investigators, there's a long history of work on the human in which instead of, pulling on muscles, and we depend on doing a kind of, you can electrically stimulate nerves in such a way as to activate some of these pathways. And then you can do sort of conditioning test combinations in order to measure the interactions among different muscles, similar to the way we had. And the wiring diagram, so to speak, that comes out of the human is somewhat different and seems to be more consistent with bipedal our bipedal posture. However, the basic patterns are actually quite similar. The problem with those experiments is that when you electrically stimulate muscles, you're activating these receptors in non-physiological ways, which you always have to consider that you're not seeing the accurate picture. But there is some correspondence. And in fact, the cat, which has been the experimental model for these kinds of studies for over 100 years, actually is remarkably close to the human, despite the fact that it's a quadruped and that we are bipeds.
Where do primates fit into? Because the primate, I would think, would be the closest to the human.
Right.
The non-human primate.
Right. There is sort of a dearth of information on these same questions in the primate for historical reasons. I guess it was decided that there was so much relevant information from the cat, they might as well just go right to the human and see what to do. So there isn't not a lot of information on the non-human primate. Okay, so yeah, really quickly because we're.
What is why would we say that the feedback is not muscular feedback but limb feedback? What is the difference?
By
The limb feedback and the muscular feedback.
what we're saying is that the feedback from a given muscle is not only distributed back to that same muscle, it's distributed widely to other muscles. So whatever it's regulating must be relevant to the entire structure and not on an individual basis. Okay, so I want to get back to this question. We talked about the fact that these pathways from spindles and tendon organs might be combined in the spinal cord to regulate stiffness of the limb. What's the evidence for that? Okay. So how do you actually do experiments to probe that? Well, according to the stiffness regulation model, as I said before, limb stiffness is determined by the balance of length and force feedback. One experiment we can do is knocking out length feedback. And it turns out that a colleague of mine, Timothy Cope, discovered that if you cut a peripheral nerve going to a muscle and immediately repair it and wait about 9 months, the nerve grows back into the muscle becomes powerful again, it can exert just as much force as it did before, but the length feedback from that muscle is permanently disrupted and not there. So people who have had transection injuries and surgical repairs often have great difficulty with dexterity, not because of the fact they can't exert forces, it's because they don't have the sensory feedback from those muscles, the kinesthesia, to execute those fine movements. So we can knock out length feedback. Force feedback is more difficult to knock out, so the way we study force feedback is to see how it's modulated under different tasks, and we'll get to that in a second. So if you actually reinnervate the triceps suri muscles in a cat and allow the animal to recover in a social colony and having a great time for a year or more, one finds that the reduction in length feedback that accompanies reinnervation results in a loss of knee and ankle joint stiffness. So this is the kinematics of walking in an animal walking down a ramp. And it's really pretty simple. When the animal puts its weight on the leg, this is all the stance phase, the knee and the ankle give a little bit when the animal puts its weight on that limb. And then the two joints extend, pushing the animal off to the rest of the stance phase. After the reinnervation, these muscles are now capable of producing every bit as much force as they did before. But now look what happens. now the yield is prolonged and sort of stays there through the entire step cycle. So there is a profound loss of ankle joint stiffness after this reinnervation procedure, which says that the pathways from muscle spindles must be important in increasing the stiffness and regulating the stiffness of the limb. To go at trying to test the role of force feedback from tendon organs in limb stiffness is somewhat more difficult because we don't have a simple way of knocking it out. But here's a couple of ideas that we've been pursuing that might have to do with how these pathways might be involved in task specificity. So one idea is, we said that force feedback is widely distributed through the limb. Let's say the force feedback was focused more on the distal musculature. That is, the distal muscles were more subject to force feedback coming from elsewhere in the limb. That would mean that the compliance of the distal limb joints, should be joints, not segments, would be reduced. And that might be useful when we're interacting with the outside world, because when we interact with the outside world, either by stepping on the ground or hitting a wall, some compliance is desirable, and those joints that are right at the interface point would be the ones that we would want to have the compliance control. But there's another argument that if we're doing something like jumping or running really quickly, we run into this problem of inertial disparity between the different limb segments. If you were running really rapidly or jumping down from onto the floor, it's those poor little distal joints that are attached to the light, low inertia limb segments that are going to be taking the brunt of the interaction. So we might actually want to have inhibition going more from distal to proximal in order to help compensate for that non-uniform distribution of inertia. These are just two ideas that we've been kicking around. So the idea is that we might have a gradient of force feedback either from distal to proximal or proximal to distal that might be useful for different tasks. And Kyla Ross was actually a student, Steve DeWorth and mine. She was in the first class of biomedical engineering students at Georgia Tech and Emory, right? I think it was the very first class. Yep. And so Kyla did some experiments in my lab, and she used the locomoting decerebrate cat preparation that I was talking about before. And she, this diagram just shows different muscle groups. This is the quadriceps group, these big knee extensors up here. The triceps suri again, the ones down here in the shank. And these muscles controlling the toes. And we can summarize her findings by saying that during slow walking on the treadmill, that the force feedback, which is shown by all of these arrows, was strongest from proximal to distal. So under those conditions, it seems to favor that first hypothesis that under some conditions, a proximal to distal gradient of force feedback would be more desirable. We still haven't gotten to the stiffness part yet, but at least that's a good start. The red ellipse here just refers to the fact, and I've been sort of hiding this a little bit, but I did mention at the beginning, these particular muscles here also showed a transition from inhibitory force feedback to excitatory force feedback. So under these conditions, the two kinds of force feedback were coexisting, and I won't go into that in any more detail unless you would like to discuss it during the discussion. So Mark is a champion of this other idea, right? And that is that, again, referring to the fact that there's a disparity in mass of the different segments of the limb. What Mark has been doing is noticing that in our last few years of experiments, actually going way back to Bonacera and Wilming's work, is that in some of these animal preparations, and these preparations are reasonably consistent, but they do vary in terms of which pathways seem to be active at different times. And we can see patterns, both of those distal to proximal and proximal to distal gradient patterns in different preparations. And what Mark is trying to do is actually to document those, to find out what the patterns are, do they correspond to those two kinds of gradients, and to see what happens. Trying to design experiments to see what would happen under conditions where the animal might take advantage of this distal to proximal gradient of force feedback. Again, I'd love to spend more time on each one of these topics, but I need to move along here. Finally, Iram Niazi is a graduate student in applied physiology. She's a physician from Pakistan who's doing her thesis here. And she is collaborating with Dr. Dina Howland, who is an eminent researcher at the University of Louisville, who studies spinal cord injury in cats. She does partial spinal cord injuries on these animals, so they're actually able to move quite well and take care of themselves. But what she finds is they fail on certain motor tasks, or they don't perform as well as an animal without the spinal cord injury, and that gives her a way of evaluating the different treatments that she is using that involve a promotion of regeneration of the spinal cord and also the use of drugs to prevent scar tissue from forming. And so Iram has actually just completed her thesis experiments, and she found that following spinal hemisection, force feedback is greatly amplified. It's much, much more powerful than in any of our control experiments. And it's strongly polarized from distal to proximal. Regardless, every single animal seems to behave that same way. So after spinal cord injury, it seems that this force feedback system is greatly distorted. And this may account for the problems of balance and stability of these animals. As I said, they actually can manage quite well. If you see them walk across the floor, they may look reasonably normal. A little shaky maybe, but if you put them on an unstable surface, they have more trouble than a normal animal. So we think that this greatly exaggerated force feedback may be part of the problem. Finally, the last little bit here in the last couple of minutes, Ginger Gottschall was a postdoctoral fellow in my laboratory, is now an associate professor at Penn State University. And she was interested in the vestibular system. And I won't go through this again in detail, but we had observed that animals walking, or not we, famous scientist Judy Smith from UCLA had observed, and her colleagues had observed, that when animals are walking uphill or downhill, they use a completely different pattern of muscle activation pattern. So something in the spinal cord has to change in order for the animal to adopt these different patterns because the physics of walking uphill and the walking downhill are entirely different. Basically, when you're walking uphill, you use your extensor muscles for propulsion. When you're walking downhill, you turn those propelling extensor muscles off and you use your flexor muscles as brakes. So you kind of switch the role of different muscles. And what Ginger managed to find was that combining the feedback from the vestibular system and feedback from the afferents, the muscle spindle afferents in the neck, there is generated a body orientation signal which automatically causes the switch in these patterns of movement. Now, what does this have to do with stiffness and all of that I've been talking about? Well, we predicted that during the downhill locomotion, you might want to have an somewhat amplified inhibitory force feedback, particularly focused on the distal joints, in order to use your muscles more as brakes than as propelling devices. So my senior graduate student, Chris Tuttle, embarked on a series of experiments to use the same experimental paradigm as Ginger had used for her locomotion studies to see if an animal was in the so-called downhill versus uphill mode, whether the stiffness of the limb would be affected, and if the stiffness of the limb were affected, what are the underlying mechanisms. And in short, he connected the limbs of these animals to a robotic system which perturbed the limb in different directions, and we could measure endpoint stiffness, which is represented by this ellipse here. He measured both the sagittal plane ellipse, which is in the plane of the screen, and the horizontal directions of stiffness, which are perpendicular to the screen. And what he found was indeed that in the downhill condition, the animals indeed showed a reduced stiffness both in the sagittal and the horizontal plane. And I won't show you the data because it would take too much time. He also found, using those muscle stretching experiments that I talked about earlier, that in fact this reduction in stiffness was correlated with increased inhibitory force feedback. So we think that this force feedback system is very, very important in the regulation of limb mechanics. The final thing I wanted to mention is that Gareth Gouvanison, who is a student of Steve DeWorth's, Steve's not here, and mine, who is an engineer, who is actually designing a microelectrode array that fits on a muscle that can be used for stimulation and recording of that muscle. And this is for rehabilitation purposes. In other words, to provide more strength of contraction in muscles in patients who are either weak or paralyzed. Alison Cloutier is a Master of Science in Prosthetics and Orthotics student, and for her capstone research project, she's working with Gareth to test whether electrical stimulation of muscles also produces normal or natural inhibitory force feedback to other muscles, to see that if you put such a device into a patient, if it would actually restore some of the normal proprioceptive pathways that we have. So conclusions, part 2, removal of length feedback reduces joint stiffness. Modulation of force feedback may be an important mechanism for regulating limb stiffness. And the postural dysfunction after partial spinal cord injury results in part from an amplification of inhibitory force feedback. This work has been supported by mainly 3 grants spanning 1983 to the present. I have a number of colleagues that have really helped with this. Tech support from these 2 individuals. Alex Bragg was an MD Ph.D. student, now a practicing neurologist at Emory, who actually wrote some really important software for us, and a whole host of students that I didn't mention during the talk. Thank you. Stunned silence. Bob.
In terms of differences between uphill and downhill, I couldn't, my eyes are probably not good enough, see what the angle was, and I wonder if we're only slightly perturbed from level, if we get a different result than if we're significantly different than level.
Right. Well, that's a really great question. So the question is, does the actual slope that you're walking up and down change? So I have a couple things to say about that. First of all, yes, in Jody Smith's experiments, she was actually a co-worker of Bob Greger's. They showed that putting animals on different slopes, and they did use different slopes, that the changes in coordination of muscles actually was somewhat proportional to the slope. But there obviously are some behavioral differences. Cats will hold their heads in different orientations. If it's a really steep slope, they may tend to look down the slope. If it's only a slight down slope, they tend to keep their heads more level. And these have profound implications for the sensory feedback that's coming back. The other thing I wanted to say is, this is a cat, it's a quadruped. It was a great animal to study this particular behavior. And so you might say, well, what relevance does this have to humans? Because humans are bipeds, and we walk up and down hill. We don't go like this, right? So it turns out that we actually do change our trunk posture slightly when we go up and down hill. And a student of Bob Greger's, Andrea Lay, who is an engineering student here at Georgia Tech, showed that when humans are walking up and down hills of different slopes and different directions of slope, that the muscular activation patterns that we see in the human are actually remarkably similar to those in the cat, and it probably comes about because there are slight changes in trunk posture which engage the same sensory mechanisms that we saw in the cat. And Ginger has actually followed that up at Penn State, so that's maybe more than you wanted to know. But Okay, well, thank you very much.
I'll ask one
Oh,
more question.
okay.
So what are medical students being taught now? Are they being taught more or less the state of our understanding?
No. They're not. They're taught. I can list a whole host of basic concepts which are basically outmoded, like the upper motor neuron syndrome is one of them. The clasp knife reflex is another one. The myotatic unit is still being taught as a bastion of, I mean, this is
Well, you
what
made
students.
that comment at the beginning.
Yeah, right. It's,
I hope that doesn't mean that all of medical education is 20 years behind the times.
well, I think when we start making more progress on treating spinal cord injury and things like that, that information will become updated in the clinic, I hope.
Yep. Okay.
Thanks.